Hello, my name is Lixin Yang, and I am a residential student at NCSSM. This summer, I participated in the Glaxo Summer Research Program led by Dr. Monahan, where I looked into inhibiting a specific DNA repair pathway in an attempt to improve chemotherapy rates. All of the healthy cells in our body have a multitude of DNA repair systems to help mitigate against DNA damage from everyday exposure, such as UV rays from the sun. However, if cells become cancerous, many of these healthy DNA repair systems are lost, and they have to resort to an alternate repair strategy called translesion synthesis, or TLS for short. TLS is not very accurate due to its ability to bypass DNA lesions to continue with cell replication, and tumor cells who use this type of repair system often develop new mutations that become increasingly harder to treat with today's chemotherapy treatments. Inhibiting TLS would mean a higher success rate in chemotherapy and other cancer treatments, limiting the number of mutations and relapses that tumors develop. So I looked into the specific processes of TLS to try and find a way to inhibit it. In translesion synthesis, the main polymerase used is Rev1. There are two pathways that can start TLS, the Rev1 dependent pathway and the Rev1 independent pathway. To fully stop TLS from occurring, I looked for drugs to inhibit each of the two pathways, and I found two small molecules, JHRE06 and T2AA. The idea is that both of these drugs would bind to a specific polymerase such that it can no longer bind to Rev1, the main protein, effectively creating a synergistic effect and inhibiting the TLS process on both pathways. In order to see if my hypothesis works, I carried out an experiment using Arabidopsis thaliana, a type of small plant, as my model organism. The seeds were grown on MS plant growth plates, which are stocked with nutrients so that they can germinate. I first exposed 10 seeds to UV light initially to see if I can induce TLS in the plants, which would be expressed as germinating seeds in a healthy plant. After it was proved successful, I plated 120 seeds, 60 of which I put under stratification, which was a method to break seeds out of their dormancy stage in order for them to germinate at the same time. I am currently growing all of them vertically so that the roots can all face the same direction. In the future, I am working to transfer all 120 seeds to individual plates with the two drug treatments to see if there is any change in the root growth of these plants, which would signify that TLS has been inhibited. To analyze the roots of each plant, I would conduct a root bending assay where the plants are flipped upside down, and I would measure the degree of angle that the roots take as it reorients itself right side up. If TLS is successfully inhibited, the root bending degree would be a lot higher than compared to the control, as it would take longer for it to grow and thus prove my hypothesis to be correct. I want to personally thank Dr. Monahan and the rest of my classmates in our Glaxo program for their continuous support during this time, and I plan to continue this experiment through all of fall next year, and hopefully the results will be able to shed a little bit more light on the process of translesion synthesis so that hopefully it can be applied to medical research in the future.